Hey, my name is Jared. Today we're going to be going over the sarcomere, kind of the anatomy of it, as well as how a muscle contraction works, what chemical processes happen to make that possible so that our muscles actually work. So let's look at the sarcomere. This diagram is essentially showing us all the filaments that work together to create a contraction. Our two major components are myosin and actin. Myosin is going to be found in the middle, and these are thick filaments, and they have myosin heads that stick out of them. And then our actin filaments will be found on the ends, kind of, and they're thinner than our myosin filaments. What's anchoring these two other sarcomeres will be a Z line, which is composed of cap Z and alpha actin, essentially tethering all your actin filaments and your myosin filaments to one point so they can actually interact. This kind of V shape here is titin, that binds to myosin, keeping it in place. In the very middle, you'll have an M line where there's kind of a switch in the direction of myosin heads. And that's a distinct feature you can find on imaging of a sarcomere. You'll have an A band, which is composed of where the ends of the actin filaments meet the myosin filaments. H zone does not include actin, but includes myosin only. A band's a little bit darker than our I bands. I bands only include actin. A bands include myosin and actin, but because myosin's so thick, it'll appear darker. And yeah, so that's your basic makeup of what a sarcomere looks like and how your components are interacting. So let's turn our attention a little bit more to our actin filaments. First off, kind of at the core, you'll have a nebulin strand, which goes throughout the whole actin filament. And it'll be anchored to a cap Z at one end and a tropomodulin at the other. Usually tropomodulin is found closer to myosin, and cap Z is where you anchor into alpha actin. So actin is made up of smaller components that bind together, and those are called G actin. And so G actin is kind of like if you think of pearls on a string, G actin is essentially those little pearls. And then F actin is when those pearls line up and are kind of polymerized along in the strand. So if you look here, I've kind of drawn two strands of F actin that crisscross. And those are your linked circles here. And then there's another line that overlays the F actin, and that's tropomyosin. And I think I mentioned that G actin has binding sites on it, and that those binding sites will be available for myosin to bind later. But tropomyosin will cover those binding sites so that myosin can't bind when it doesn't need to. Another component of actin that is very important are these troponin molecules, and they kind of have three components to them. They kind of look like Mickey Mouse right here. And if we go up here to this corner, troponin will have three components to it. You have an I component, a T component, and a C component. I will attach to actin, to these F actin film or G actin, kind of anchoring it to the actin itself. The T component will be bound to tropomyosin, that overlay that covers the active sites. And then the C component will bind to calcium, which will be important in changing the conformation of of changing the conformation of troponin such that we can pull tropomyosin off of the active sites. Another thing to keep in mind with the C component of troponin, has 4 binding sites for calcium. 2 are high affinity, 2 are low affinity. So in a scenario where you have a high concentration of calcium, the low affinity binding sites, the low affinity binding sites will have calcium in them. The high affinity binding sites for calcium most of the time will have calcium attached because they don't want to let go of calcium. Okay, we've covered actin as well as how it works with troponin to open up binding sites to make it available for myosin. So let's move on to myosin. Myosin has myosin heads attached to your main filament, and they'll come off of that main filament at a 90-degree angle, and these myosin heads will have a hinge region. That's where the 90-degree angle comes in. And the heads will have actin binding sites. That's why it's important to uncover the actin binding sites for myosin to uncover the binding sites on actin so that myosin can attach. And then there will be ATP binding sites on the myosin heads, so we can cock and uncock these hinge heads essentially. Okay, so let's kind of look at how that cocking and uncocking action happens. In a cocked position, and that's where you're at 90 degrees, you're not attached to actin at all because tropomyosin has covered active sites. You'll have ATP bound to your myosin head. Then ATPase is going to hydrolyze that ATP to become ADP and phosphates. At this point, it's still attached to the myosin head, and we're still in a high energy phase for this myosin head. So it wants to attach, it's ready to go, but it can't get to active sites yet. It just needs tropomyosin to move it, and all that energy will be let off, but it can't do that if the active site is covered. So what's going to happen, say tropomyosin moves, we bind, that binding energy is going to release into ADP and phosphate, and once ADP and phosphate unbind from the myosin head, our myosin head will uncock, causing us to go through a power stroke. And that's where you're bound to the actin filament, you're pulling it across the myosin filament. And so they kind of slide across each other. If this is myosin and this is actin, they slide across each other. So now we're uncocked, we're at a low energy state. We need to get back to a high energy state, and the only way that happens is if we get ATP back attached to the myosin head. Once ATP attaches to the myosin head, it will release from the active site on G-actin, and then we've reset. Something to keep in mind here is if G-actin if there's calcium in here at all that is pulling tropomyosin off of the active sites, then we can go through the cycle several times so we can continually pull the actin and myosin filaments across each other to actually get some distance, because if we only pulled it once, we're not going to move the muscle very far. Okay, so that's kind of how myosin and actin interact so that our sarcomeres can shorten, not shorten, but so that they can contract, causing the whole muscle to move. But let's consider how this all works from our neurons all the way to the contraction component, right? So you have a neuron here, and we're sending an action potential down this neuron, and it's depolarizing. Once you get to the axon terminal, it's going to be depolarizing, and then that's going to allow voltage-gated calcium channels to open, allowing calcium into the axon terminal. That calcium is going to play a vital role with snare proteins. Those snare proteins are going to bind to acetylcholine. Acetylcholine is then going to be exocytosed out of the axon terminal into the synaptic cleft. And then acetylcholine is now in the synaptic cleft, and it is going to bind to a ligand-gated channel on our sarcolemma. Sarcolemma is a membrane that surrounds all of our sarcomeres, and so you bind to a ligand-gated channel on sarcolemma, that membrane, that opens up a channel so that sodium can get into this intracellular space, and which generates an action potential down the membrane of the sarcolemma. And as that depolarizes, you propagate and have more sodium potassium ion channels opening, so your action potential travels down the sarcolemma membrane. Part of the sarcolemma membrane has its T-tubules, and these are invaginations that allow this action potential to travel down to a point where they're closer and surrounding the sarcomere. And in these T-tubules, you'll have what's called dihydropyridine receptors, and these are calcium ion channels. But even though with the depolarization they do open, they're not letting calcium into the cell. In fact, they act more of a receptor, or more of like just a sensor, so that this dihydropyridine can now stimulate a ryanodine receptor, which will allow calcium to move into the intracellular fluid. So we have on the T-tubule, we have a dihydropyridine receptor, and then on a sarcoplasmic reticulum, you have a ryanodine receptor. That sarcoplasmic reticulum contains a lot of calcium, and it's intracellular, or it's going to allow the release of calcium into the intracellular fluid, and that calcium, as discussed before, will bind to troponin and cause the whole contraction phase to occur. And I think that should be most everything. A couple other things would be, you have a motor end plate, that's where the sarcolemma and your neuron meet. And the sarcoplasmic reticulum kind of is like a webbing, like a sleeve over your sarcolemma, so you can get calcium to all parts of the sarcolemma, or of the sarcomere, very quickly. And yeah, that's how sarcomeres work, how calcium plays a vital role in their contraction, and how actin and myosin work together to make that contraction possible.